expression level, it's green. If it's yellow, it's unchanged. So rapidly scanning this with a laser, we can cover 43,000 different gene probes and say which genes go up and which go down consistently. We do everything in replicate. So we can cover the whole human genome. So we can do this, say, for a patient. Let's just say this green patient here. Between our internal control, here's our seizure area, here's our non-seizure area. Say we find 500 or 1,000 genes out of 43,000 that meet our statistical cutoffs. We say, that's great. But does that mean anything for the next patient that comes along? So we take the next patient that comes along, we do the same experiment. And we take the next and the next and the next. And even with as few as 5 patients, if you have anything left in this intersection, it may suggest some common pathophysiology, some patterns of genes that reflect that seizure onset area, the part of the brain produces epilepsy. So from 43,000 genes in the study, with 5 patients, we simplified it to 137. Now 137 genes is still a lot of genes, but in big data it's really not. And you can actually almost just go to PubMed and look at all these genes and kind of try to figure out what they do and make some patterns analysis. So that's what we do. It's called ontology and bioinformatics. There are programs that do this and say, what are the common pathways represented by these 137 genes? What do we know, and statistically, what are the most, have the most impact factor? And one of them is a kinase family called MAP kinase. So we build a model then from the epileptic activity that maybe this signaling pathway, MAP kinase, may have some role. We don't know if it's causing it, resulting from it, but statistically, it kind of is our low-hanging fruit of just jumping out at us as something that we didn't know before that now we think is important in human epilepsy. We can look at things called promoters, which are the parts of the DNA that drive the expression of genes. One of them is called CREB. It's a transcription factor that binds to DNA and turns on at least 40% of these genes. It's known. And there are many, many transcription factors, so that's probably not by random chance that 40% of the genes are turned on by CREB. And one of the pathways that's known to turn CREB on is MAP kinase. So again, we can build this into the model. So the idea is we take the 137 genes and we build a hypothesis, a model, we call our low-hanging fruit model, of what is overrepresented in the difference between the tissue that seizes and the tissue that doesn't. And this is the pathway, and it looks very much like synaptic plasticity and memory in the brain. You can say, well, that's not surprising. I guess the brain is firing all the time. You're going to turn on these pathways and genes. But we didn't know this before. So what do we do with this pathway then? Is it interesting? Do we stop? Do we run on to the next thing? We need to validate. So in order to get something back to our patients, whether it's a biomarker for a test or a diagnostic or a therapeutic, we need to prove cause and effect. We need to show that these pathways are truly present both in human tissues, then maybe go to an animal model and prove that if we knock it up or knock it down, it actually has an effect on the disease, so that then we can go back and develop drugs for human trials. So this is the sequence we're going to talk about a little bit of how we then translate this finding and understand what this means from just statistically looking at everything in the genome and narrowing it down to this low-hanging fruit. So let's talk about validation. Remember, at each block of tissue, we don't only collect tissue for RNA, which we do for the microarray studies, but we also look at protein. So protein, you can do a test called a Western blot. Western blot looks at proteins. This is a protein in the MAP kinase pathway called phosphor-ERK. When it's activated, it becomes phosphorylated. So this is phosphorylated ERK in the epileptic tissue. This is the nearby tissue that's non-epileptic. It's not phosphorylated. So this is actually a physiological process predicted from the gene expression that we validate on a piece of tissue, right in the same piece of tissue. Here's that transcription factor CREB. When it gets activated, it also gets phosphorylated. So the fact that this band is dark here and light there means that it's activated again in the epileptic tissue. And here is a control. This is the non-phosphorylated. It's present, but you look at the ratio. So very quickly, we can validate the pathways that are predicted mathematically from the gene expression into pathways that are truly overrepresented and expressed in those tissues. And then we can build a model. So from this first experiment, we do see evidence for common pathways linked to these MAP kinase, transcription factors, and basically something that changes the wiring, the synaptic plasticity of the brain. So next question we started to ask is, well, that's the areas that produce seizures. We know that there's areas that spike, and sometimes spiking and seizures go together, sometimes they don't. So this was a larger project. Instead of 5 patients, this was a 15-patient series. Different age, different sex, different other types of diagnoses. And all we did was do our internal control of an area of that brain that had a high frequency of spiking and one that had less. So in this case, it was 6 versus 0. But another patient, it was 576 versus 157. These are spikes per 10 minutes. So one could argue that there's so many differences in this pattern. When we do our Venn diagrams, we're going to find absolutely nothing at the intersection of these 15 circles. In fact, when we did this experiment and used the same statistical cutoffs, now our N is higher, so it gives us a little more statistical power, we got 1,700 genes. So it's actually larger. So these are our seizure onset genes. These are our high spiking genes. What's interesting, if you look at the overlap of these two groups, is there's a lot shared, and we know areas that spike also seize, so it kind of makes sense. But also from a mechanistic point of view, there are clear areas in this circle here that don't overlap. So there may be pathways, genes that produce seizures in the human brain that are different than those that produce spiking, which has their own sets of genes. So we can now segregate some of the electrical activities to different populations of genes, different potential pathways that we can go back and try to understand. So what else can we do? So this is what we call bigger data. We can take our data and we can ask a question: what does your DNA look like on epilepsy? So the genes are in different places on your chromosomes, and if you simply plot them, the 1,700 genes, the spiking genes on your chromosome, where are they distributed? Are there certain parts of our genes that light up? And again, a lot of these genes are human brain plasticity genes. So it's an interesting question to look at the organization of our thinking genes on our chromosomes. And so this is the first demonstration of what your DNA looks like on epilepsy or with lots of activity. These are different chromosome numbers, 1, 2, 3, 4. And where you see a peak, that's a cluster that would not be expected by chance or concentration of genes in that area that kind of light up. So we call these hot spots. So in your DNA, in the epileptic tissue, there are certain parts of your DNA that either turn on more genes or in some case turn off populations of genes, which is kind of interesting that we have these 33 hotspots in our chromosomes. So how the genes are organized in our DNA may have something to do with plasticity in the brain. We can then look at these hotspots and link them to evolution. Some of these are very human specific, which is quite interesting. We can also look at them in relation to diseases because we've mapped certain diseases to different chromosomal locations as well. So this is kind of the fun stuff you can do downstream once you have these data sets to understand what they're doing. Another question we like to ask is, what does epilepsy look like? And we also call this tissue biomarkers. So this is the picture I showed you, and actually I didn't give it credit. I actually stole this picture from a pharmaceutical company. It was a poster in my clinic. When I talked to my patients, I pointed to this and I said, here's your focus and here's how it spreads. But this little bullseye here, that's kind of the state of knowledge of what we know about what an epileptic focus looks like. But is there a way now we can use these pathways and genes that are highly represented in areas that produce spikes and seizures to get a better idea what parts of the brain, which layers, which cells are actually involved in these epileptic circuits? So here's that pathway. This is our low-hanging fruit pathway from MAP kinase to plasticity genes. If you look at this same antibody that stains diphospho-ERK, it's only on the most superficial part of the human brain. And you don't see this, obviously, in the areas that don't spike. If you zoom in, you can see that the protein is expressed in all these synaptic areas and in the nuclei of the cells. And if you see these little holes here, those little holes in the brain, those are the locations of the cell bodies. So if it's signaling to the nucleus of the cell to CREB, which is the transcription factor, you might expect to find CREB turned on there. And in fact, it's turned on in this narrow band of cells in layers 2 and 3. We have 6 layers of the human neocortex. These pathways are really restricted to this narrow band just below the surface layers 2 and 3. So all of a sudden, from 43,000 genes to 137 to a pathway, we've now narrowed down the disease, at least the spiking of the disease, to this narrow band of neurons in layers 2 and 3 of the cortex, which is kind of fun. And here you can see a higher magnified view of this area here. You can see the circles here is because this transcription factor is only in the nucleus of cells. And then we also start to see interesting things like it shuts off when it goes down into the deeper recesses of the sulci of the brain. We do lots of control experiments because we don't believe anything we do because it's all uncharted territory. This is showing that the non-phosphorylated transcription factor CREB is in every cell in the brain. You can see here, but the activated transcription factor is only in this narrow band here. We then can go downstream and look at some of the other pathways, such as this immediate early gene. This is an in situ showing the genes turned on there in the same layer, and plasticity pathways. Again, all turned on in that narrow band in layer 2 and 3. So this entire low-hanging fruit pathway seems to be very precisely localized. And from an engineering point, if you're going to make a device to go after and you wanted to put something into the brain and shock it or do something, we actually know where to go now from these tissue biomarkers. So from electrical activity to genes to pathways to localizing in the brain. So why layer 2 and 3? Now we have to start thinking about wiring diagrams and what's the normal wiring diagram of the brain and what goes wrong in epilepsy. So we know in the 6-layer cortex, a lot of the information subcortically comes up through the thalamus to layer 4. A lot of the outflow, the big cortical neurons, let's say make my arm move, they're in layers 5 and 6, all deeper layers. We're talking about layers 2 and 3. What do layers 2 and 3 are known to do in the brain? They're actually known to form lateral connections. So when this part of the brain wants to talk to this part, you connect through layers 2 and 3. And why this is really exciting is because what are we talking about as synchrony? Synchrony is huge, huge sheets, 10 square centimeters of brain firing synchronously. Maybe it makes sense that this is happening in layers 2 and 3, which is the normal part of the brain that forms lateral connections. So how does it get there? So switching to another story. And this is fun. When we published this study and we put out a little press release, we got more hits on math sites than we did in brain sites, because mathematicians like this study. So we call this the cellular interactome. So one of the problems with epilepsy is many diseases, many other things can cause it. You can have a stroke and have epilepsy, you can have a brain tumor and have epilepsy, you can have a developmental abnormality, you can have an infection. But when we actually look at the part of the brain, when we pull them out and look under the microscope of the part of the brain that actually produces the seizure, it looks pretty normal. So this is an area of the brain, a human brain. It's stained with an antibody to something called glial fibrillary acidic protein, which stains scar tissue in the brain, glial cells. And this looks pretty normal. All layers, 1/6, nice. We show it to our pathologist and say, look, this patient's seizing from there. What do you see abnormally? Looks fine. So what's wrong with this? So how can we use big data to answer that question? So going back to these really statistically significant 1,700 genes that from the 15 patients. This is Fabienne Dachet's work, who's here with me in bioinformatics in my lab. And these are 30 different human brain samples that have different levels of spiking. Some have high spiking, some have low spiking. And this is expression of a gene. So again, the more copies of a gene, the higher the expression level. So this is gene 1. You can see there's many copies, the high expression in these samples on this side. On this side, there's low expression. So there's a certain pattern of gene expression for the different blocks of tissue. If you randomly look at another gene in those tissues and say, well, what's gene 2 look like? What's its pattern across these 30 different human samples? Very different. So it's low here and high here. So it's like these genes are not regulated, they're not coordinated in any way, they have nothing to do with each other. Now if you look across the 43,000 that we actually study at all these different patient samples, you can find amazingly tight clusters. With 95% confidence, these clusters of genes, hundreds of genes, go up and down together as groups. So I think of it like a school bus full of kids. Sometimes the school bus goes to the zoo, sometimes it goes to the park, sometimes it goes to the museum. But if you could actually identify all the kids in that bus, you could probably figure out which school they came from. And that's kind of what we then had to try to do. So we found clusters of genes that are co-expressed. We postulated that these are different cell populations that are changing in these tissues as a function of the epileptic activities. So randomly we say, okay, this is cell population A, cell population B. So now let's take cell population A, and let's take the names of those genes shown here, and let's do a PubMed search for each of these genes versus all the different types of cells that could be in the human brain. So here's an astrocyte, an endothelial cell, and basically do an iterative PubMed search and look at all the abstracts. And if we see the same 2 words in an abstract, we say we got a hit. And if you do this across the whole group, you can see we get more hits with the word endothelial in this group of genes than any other. So endothelial cells are blood vessels. So the prediction is, since these genes go up, that if we were to stain the tissue and look at it, we would see more blood vessels in the part of the brain that produces seizures and spikes than a nearby part that does not. So this is how we make, from mathematics and statistics, we make predictions. And of course, I'm going to show you we validate every prediction that we make. So this is, you guys know Cytoscape program. It's a graphical, visual, analytic way of actually looking at these relationships. So here's this cluster of genes, the endothelial cells. The closer these genes are to each other, that's the tighter the correlation that they go with each other up and down together. And not surprisingly, the other predictions nearby is a red cell and a white cell. Well, why would these cells go up in a near vicinity to endothelial cells? Because blood vessels are full of red cells and white cells. So You would expect that. But we also found 4 different neuronal populations, 2 different populations of cells called microglia, which are inflammatory cells in the brain, and then 2 populations that actually go down. So all of these go up, these populations of predicted populations of cells, except this one called an oligodendrocyte and a neuron type 1. So this is like fun and fun with math, but is any of it really true? Before getting that, the other thing you can do is you can take the eigenvalue of each of these clusters and again look across your 30 brain samples and see how they change relative to each other. We have some idea based on the locations on the cytoscape. But this is a dendrogram showing, not surprisingly, the endothelial cell and the blood cell. They go up and down together beautifully. So as you would expect, when you have more blood vessels in that piece of tissue, you're going to have more blood in that tissue. So that's a nice confirmation. But then you get some new things you never thought of, like this population of inflammatory cells, microglia 1, and this mirror image of neurotype 1 that goes down almost in opposite proportions. So now you can kind of think that maybe we can actually start building models of what's the cellular abnormality that we couldn't see when we just routinely stained the tissue based on this type of analysis. So here's an example of 2 different populations of these inflammatory cells, microglia, it's shown in the green lines. The fact that there are 2 different green lines that have different shapes means that we probably have 2 different populations of these inflammatory cells. One that seems to change somehow related to the blood vessels, and one that changed in regard to these other populations of neurons. But in both cases, microglia are increased. So we take the tissue from the exact same block where we predict we're going to see